Today we will be talking about antibodies, immunoglobulins. Right? The concepts about immunoglobulins are very important. These are one of the most important molecules in our body. We cannot survive without them in such a hostile world where there are so many microbes and so many pathogens which are trying to invade our body and destroy our tissue. Now, exactly what are antibodies? Antibodies. They are also called yes please, immunoglobulins. Now, what are exactly antibodies? From where they come in our body and exactly where they are present, right? Major portion of the antibodies circulating in the blood, right? Antibodies are special protein molecules. Antibodies are very special type of protein molecules which are secreted by plasma cells. Antibodies are special type of protein molecules which are secreted by the plasma cells. Now, what is a plasma cell? Actually, whenever a B lymphocyte becomes fully differentiated and becomes fully functional, right, it starts producing antibodies. It starts secreting Y-shaped molecules or antibodies. So, antibodies are the products of proteinaceous product of well differentiated B cells or protein products of plasma cells. Is that right? Now, exactly in the blood, in which component of the blood antibodies are present? Yes, please. Question goes to very impressive Dr. Safa. In which component of the blood antibodies are present? Let's come with very simple thing. Everyone knows that blood has two components, cell and, yes please, plasma, and plasma has many components. Is that right? One of the component is plasma proteins. Actually, antibodies are the part of the plasma proteins. That's a very basic concept, that where antibodies are. Antibodies are present, yes, present in your blood as the proteinaceous component of the plasma. Now, plasma has many type of proteins. Now, we have to see that within the plasma and within the plasma protein where antibodies are exactly present. So, Dr. Israel may answer us that your plasma has proteins and one component of the plasma proteins are basically antibodies. Now, which component of the plasma protein is having antibodies? I'm about to be impressed by him. I think he is very serious right now. He doesn't want to answer. So, I will do it myself. Look, one of the way, let's suppose that on this filter paper, we put a drop of plasma, right? We put all the plasma proteins here. We put all the plasma proteins here. You know, plasma proteins are positively charged or negatively charged? You like to keep secrets, you don't tell me. Plasma proteins are negatively charged. Is that right? At body pH, which is 7.4 by the way, at the body pH, plasma proteins are negatively charged. Now, because most of the plasma proteins are negatively charged, if you put a drop of plasma on this filter paper and you run the current across it, for example, you put negative electrode here and positive electrode here, of course, plasma proteins will move from negative electrode towards positive electrode because plasma proteins are themselves negative. So, they will be repelled by the negative and of course, they will be attracted to the positive. So, plasma protein will start moving. Now, as plasma proteins start moving, they segregate into group because all the plasma proteins don't move right with the same speed on this paper. You can answer this. If plasma protein is very heavy, right, that will move fastly or slowly? Slowly. Actually, those plasma proteins which are more negative and smaller in molecular weight, they will be more smart and they will rush fastly. So, what really happens that some of the plasma proteins will reach here, right? Then there will be some here here and here and here. Now these are the plasma proteins which are smallest in molecular weight, right, and they are more mobile, right, so they will come over here. This was the movement, right. Heavier plasma proteins will be left behind. Is that right? If you divide the plasma protein or segregate the plasma protein by this way, this is called process of electrophoresis. Have you heard of it? Process of electrophoresis. When you do electrophoresis of the plasma proteins, right, the electrophoresis. Is that right? Now, electrophoresis, when you do, the plasma proteins, the smallest molecular weight will be here, and as the plasma proteins become more and more heavier, they are segregated in a different band, right? Actually, these plasma proteins as a group are called, just please, albumins, because albumins are the smallest molecular weight, and this whole group of plasma proteins, these three, this is called globulins, and this is post-fibrinogen. Is that right? Another way to show this is that you draw a graph. Graph is that if you put molecular weight here and you are classifying the plasma protein according to the increasing molecular weight on this side and plasma protein concentration on this side, then this is albumin. What is this? This is albumin, and these are globulins. Am I clear? Now, out of this, this is alpha globulin, these are beta globulins, these are gamma globulins. It means primarily plasma proteins are albumin and globulins can be separated. Globulins are globular protein, can be separated, right, according to their molecular weight into alpha globulins, beta globulins and gamma globulins. Is that right? Am I clear? Albumin's molecular weight is about approximately 70 kilodalton, but globulin's molecular weight is from 90 to 120 kilodalton. Right? Now, within this shade, where exactly antibodies are present? That is the question. Now I think you should be intelligent enough to guess. In this band, this is albumin, alpha globulin, beta globulin, gamma globulin, and exactly where the plasma proteins are segregated. Israel, Dr. Safa.
Is it alpha and beta?
Of course, they are not alpha and beta, they are in gamma. Actually, if you study these plasma proteins, these are antibodies. If you study in this band, this is antibody. So, where are the plasma protein in your body? Plasma proteins are of course in the plasma, but within the plasma protein, where are the antibodies? Antibodies are part of the gamma globulins. Actually, that is why antibodies are also given another name. What is that name? Gamma globulins. Simple as that. Am I clear? So, there are three names. The antibodies, they are immunoglobulins, these are gamma globulins. Now, these are three same name for the same type of molecule. Is that right? That gamma globulins mean those globular proteins which are part of the gamma fragment, right, or gamma band on electrophoresis of plasma protein. Immunoglobulins mean what? Those globulin proteins which have immune function. They are also antibodies. And what are antibodies? Antibodies are those protein molecules which are secreted by well differentiated B cells called plasma cells, and antibodies specifically, this is very important, antibody molecules specifically react with the antigens which stimulated the formation of those antibodies. So what are antibodies? Antibodies are protein molecules secreted by the plasma cells which react specifically with those antigens which elicited their or stimulated their formation or stimulated their synthesis. Am I clear? No problem up to this? Now, if I give you a statement, I say that antibodies are those globular proteins, right, which are produced by the plasma cells, and they react with specifically with the antigens which stimulated their formation. Plus, I can say antibodies are also called immunoglobulins, because why we call the antibodies immunoglobulins? Because antibody molecules are just proteins which are part of the globulin components of the plasma protein. And which component of the globulins? Gamma globulins. Is that right? So next time, if someone asks you that in your blood where the antibodies are present, you say remove the cells, left is the plasma. Out of the plasma, separate the proteins, run the proteins electrophoretically and find the gamma band. And all your antibodies will be present in gamma band. Am I clear? There is no problem up to this. Now we come to really basic structure of antibody. As I told you, these are basically proteins with little carbohydrate component. And when proteins have a little carbohydrate component, we call them glycoproteins. When proteins have a little carbohydrate component, what we call them? Glycoproteins. To be more specific, antibodies are glycoproteins. Antibodies are glycoproteins. Now let's come to the basic structure of a typical antibody. Yeah. Antibody is basically a tetrapeptide. Any antibody is basically a, number one condition is, it is a tetrapeptide. When I say that antibody is a tetrapeptide, it means how many peptide chains it has? 4. It is tetrapeptide consisting of 2 heavy chains and 2 light chains. A typical antibody molecule is a tetrapeptide with 2 heavy chains and 2 light chains held together by, held together by disulfide bond. Disulfide bonds. So again, what is antibody? Antibody is a tetrapeptide. Antibody is of course glycoprotein molecule, right? It is a tetrapeptide consisting of 2 heavy chains and 2 light peptide chains. All of them are held together by disulfide bond. Am I clear? And it is usually a Y-shaped molecule. Let me make you a typical antibody molecule. Antibody molecule should have 4 peptides. Okay, I will make 2 heavy peptides here. Why we call this peptide heavy peptide or heavy chain? Right, this is a heavy chain and there must be another. There must be, yes please, another heavy chain. So antibody has 2 heavy chains. With that, it has 2, light chains. It has 2 light chains, and there are 2 heavy chains and there are 2 light chains, and they are held together by, yes please, disulfide bonds. These are the disulfide bonds which are holding them together, and of course they should be held together by disulfide bonds. Now let's go back to the definition. Antibody is of course an immunoglobulin which is secreted by plasma protein present in the plasma gamma globulin component, right? Antibodies are basically tetrapeptide, the 4 peptides having 2 heavy chains and 2, these are 2 heavy chains and 2 light chains held together by disulfide bonds. Am I clear? This is a typical, you can say, shape of the antibody. Now, antibodies have the specific function of the antibodies, they should bind with the antigen in a specific manner, right? Now, in which part of the antibody antigen binds? Yes, you will tell me. You could just come here and put your finger in which part of the molecule antigen will bind exactly. Yes, just tell it. Hurry up.
Just the top.
I don't know. Top means
The antigen binding.
how many points are there where antigen can bind? You don't know. Just come here. You know, be brave. Yes.
Right here?
Acha, antigen will bind here. Okay.
Like in this area?
No, I give you here. This is antigen, right?
Uh-huh.
Very sad because it's going to bind with antigen. You draw it there exactly where it will bind.
Mm,
Okay, so it right over there. You were near the point, but honestly, it should be laughing here. You know why?
why?
Because from here it can escape away.
What about
Your,
here?
no. It should be in between them like here, and then it will, both chains will cooperate to catch it. You see? Antigens are not here. They will run away. Is that right? Actually, a part of a light chain and part of a heavy chain together grab the antigen. This is very important to know. This is one antibody molecule having two heavy chains, two light chains, right? And one antibody molecule, which I'm showing here, can bind with two antigens or two epitopes of the antigen. Is that right? Now, let me tell you. Actually, the antigens which are the antibodies which are directed against different antigens must be different in this area. Let me tell you what I'm telling. For example, here is one antigen like this. Another antigen is like this. Another antigen is do you think these antigens are same or different from each other? They are different from each other. And antibodies will bind with, do you think one antibody can bind with all three of them? No. Antibodies are very specific. The antibody which will bind with this antigen will not bind with the other ones. Is that right? Every antibody is very, very specific for a given antigen, a closely structural given antigen. Now, if I say that antibody which is going to react with this, it cannot react with this. An antibody which reacts with this antigen cannot react with the other two. It means this part of the molecule, this part of the antibody molecule should be different in different antibodies for different antigens. Are you understanding? For example, let me tell you that I will make a simple diagram that let's suppose this is like, now they can catch this antigen. Is that right? In the same way, now it will more specifically fit. Suppose this is antigen number 1, this is antigen number 2, this is antigen number 3. Is that right? Am I clear? Now, opposite to that, if there is another antibody and which will fit in, which antibody is directed for that antibody will have some difference. Now, do you think antigen number 1 can bind here? No. It is designed for antigen number 2. Antigen number 2 can better fit over here. Is that right? So what we see that this part of the heavy chain and the light chain is varying from antibody to antibody when antibodies are directed against for different antigens. So we say that let's suppose if we have 3 antibody molecules, one is for this antigen, other antibody molecule is for this antigen, and other antibody molecule is for this antigen, then what will happen? All those antibody molecules which are directed against different antigens should have this area with different configuration. It means this area varies according to the specificity for antigen reaction. That is why this part of the light chain, this is the light chain, this part of the light chain is variable light chain. And this part of the heavy chain also vary. So this is variable heavy chain. This component, this part of course, this is a peptide chain. So it should have here amino end and here it should have carboxyl end. The same way heavy chain should have one amino end, another JS carboxyl end. You understand this thing? Every peptide chain has on one side amino end, another carboxyl end. So what really happens that amino end of the light chain and amino end of the heavy chain, they are having a domain. Domain means a region of amino acids sequence within a peptide chain. This domain is variable. So this is variable domain of the light chain, this is variable domain of the heavy chain. Is that right? But this component, this will not vary. It is constant. It is constant. We call it constant of the light chain and constant of the, yes please, heavy chain. This is variable of the heavy chain and this is constant of the heavy chain. And if you come to the light chains, this will be what? Variable of the light chain and this will be constant of the light chain. Is that right? No problem up to this? Let me make it more clear. Let's suppose there is star antigen, there is spherical antigen and there is potato antigen. The star antigen, ball antigen, BALL ball antigen and potato antigen. Now antibodies reacting against them will be same or different? Different antibodies. Let's suppose I show the antibody for this. These are the heavy chains, all of them. I am showing in a very simple way, you can understand. Now these are three antibody molecules, but antibody molecule which will react with the star, let me show star here, which is going to react with the star. This part of the light chain will be variable and this part of the heavy chain will be variable. Of course here if on one side it is binding with the star, on other side it will also bind with the star. So the same variable which is here on the heavy chain, here also same variable will be there and variable will be there on the light chain also. Now if this molecule is going to be this ball antigen, then what will happen? Now here is the, let's suppose I put the ball. Do you think the variable regions can be same as star? No. So they will vary from in this area. You understand it? Now we come to this. For that antigen, suppose there is another antibody which specifically react with that. Of course it will have two heavy chains and just please two light chains. Right? Now this antigen is like this. Now you understand it? That basically a particular antibody will react only with the specific antigen. Every antibody does not react with every antigen. And why a specific antibody molecule react only with the specific antigen? Because antigen binding area of the antibody has variable regions. And these variable regions, right, vary from one group of antibodies to the next group of antibodies to the next group of the antibodies as antibody group change their specificity for the antigens. Right? Now come back. So this was variable domain of the light chain and this is the constant domain of the light chain. Then this is a variable domain of the Heavy chain, this is the constant domain of the heavy chain. Heavy chain has many constant domain. Uh, Constant domain number 1, constant domain number 2, constant domain number 3, right? 1, constant domain number 1, constant domain number of the heavy chain number 2 or 3. And later on we'll find that most of the antibody classes, how many antibody classes are there? There are 5 basic classes of the antibodies, right? The 5 classes of antibodies are called, can you tell me? Immunoglobulin G, A, immunoglobulin A class, immunoglobulin M group class, immunoglobulin E, and immunoglobulin D. We call it gamed. All the immunoglobulins can be primarily divided into 5 classes. I will tell you later how we divide them, on what base immunoglobulins can be divided into 5 classes. But antibodies can be divided into 5 classes. This is immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin IG. Now you understand why we call it immunoglobulin. These are the globular proteins from the globulin component of the plasma proteins having immune function and belonging to the class of G or class of A, right, or class of M, E or D. Out of this, ME, very important, you know. Me has longer heavy chain. What does it mean? That basically immunoglobulin M and immunoglobulin E has heavy chains with constant number 1, constant number 2, constant number 3, constant number 4. Right? So constant of the heavy chains in immunoglobulin class M and class E is how many? 1, 2, 3 and 4. But immunoglobulin A, G and D, their heavy chains are having only how many constant domain? Constant domain number 1, 2 and 3. But IgM and IgG have how many? 1 additional, that is 4. Is that right? Now let's review it. Antibody molecule has how many peptides? A typical antibody molecule has how many peptides? 4. 2 are heavy chains and 2 are light chains. Why light chains are called light chain? Because the molecular weight is less. Usually light chains molecular weight is about 25,000 and molecular weight of the heavy chain is 50,000 to 70,000 depending upon, there are 3 constant or 4 constant region, right? So there are 4 peptide held together by the disulfide bond, 2 light chains, 2 heavy chains in a typical antibody molecule. Is that right? Now on the amino ends of the heavy chain and amino end of the light chain, there are which areas? Variable areas. There is variable area of the light chain, there is variable area of the heavy chains. In between these 2 variable area, what is fitting there? Antigen fits. Is that right? So antibody binds with the antigen through the heavy chain as well as through the light chains through its variable domains. And then there is only 1 constant domain of the light chains, but heavy chains have how many? 3 or 4 constant domains. Any question up to this? There is no question, no problem up to this. Now, another thing, that some doctors they were doing some experiment with the antibody molecules, and what did they do? Let me tell you, suppose I draw here a typical antibody molecule. I hope you understand it, isn't it? And what did they do? That put these molecules in a flask, right? Put the antibody molecules in flask. But in the flask, in this container, after putting the antibody molecules, I think I will make the light chains color different so that you can appreciate the diagram better. These are the antibody molecules which we have put into this container, and of course they are held together by, yes please, disulfide bonds. Now what the researchers did, they added a very naughty type of enzyme. What is this enzyme? This is called papain. They added a papain to this, and they were surprised that papain could break down this molecule at a special points. Okay, I will keep my color scheme same. Right? They found that papain could cut this molecule at this point. It digested the molecule of antibody. Antibody is a protein that was digested by this papain. And papain is an enzyme which attacked this region. This is called hinge region. What is this? H-I-N-G, hinge region. Attack the hinge region and digested from this point and this point. Now antibody break down into how many components? How many fragments? Yes, question goes to Mr. Yes. How many antibodies broken into how many components? The 3 components. Look at it. One component is this, another component is this, and another fragment is this. So antibody break down into 3 fragments. So what did they do? They put antibody molecules into a container and add the papain to that, and papain digested every antibody molecule into how many fragments? 3 fragments. And these fragments which were made, out of them this fragment has, right, amino end of the heavy chain and complete light chain, right, held together. Another amino end of the heavy chain with complete light chain. And this portion has remaining heavy chains held together by disulfide bond. Is that right? Now, then after that, they added the antigen to this. What did they add? Let's suppose this is the antigen for that particular antibody. They added the antigen to this solution. And they came to know that these fragments, antigen bind with these fragments, right? But do you think antigen will bind at this point? Or will the antigen bind here? No. Will antigen bind here? Will antigen? Yeah. Will antigen bind here? Will antigen bind here? Will it bind here? But it will bind here. So in this way, the researcher found that when antibodies are put into a container and treated by the papain, antibodies will break down into fragments. Some of the fragments will be binding with the antigen and other fragments will not bind with the antigen. These fragments which do not bind with the antigen, they found they will go to the bottom of the flask. These fragments will go to the bottom of the flask, and there they will crystallize. They will change into crystals. Due to that reason, doctor said when an antibody is treated by the papain, when an antibody molecule is, again let's draw it here and here, this is disulfide bond. Here is your light chains, isn't it? So they found when they treat with the papain, antibody will break down at this point. Is that right? It divided into 3 fragments. Out of these 3 fragments, 2 fragments were able to bind with the antigens, and other fragment was unable to bind with the antigen. And this fragment went down and it crystallized. So they called this fragment of the antibody fragment which crystallizes. FC portion of the antibody. In the books you very frequently read FC portion of the antibody or FC fragment of the antibody. C originally was put for the crystallization, that this component of the antibody will crystallize once antibodies are treated with the papain. And this fragment, this was binding with the fragment which was antigen binding. We call it fragment antigen binding. So we call it FAB. So basically a classical molecule of antibody consists of 4 peptide having 2 heavy chains, 2 light chains held together by disulfide bonds and having 2 FAB fragment and 1 FC fragment, right, or portion. Am I clear? Now, actually this FC stands for crystallization, but it also should remind you that with this end there is what? Carboxyl end of the heavy chains, because amino end was here for the heavy chains. So C for crystalline, C for carboxyl end, C for complement activation. Some of the antibodies activate the complement. This is the portion of the antibody which activate the complement. This is the portion of the antibody which activate the complement. It means in this domain, more specifically, if you remember, there was for heavy chain, how many domains were there? Heavy chain, variable, constant, heavy chain 1, constant heavy chain 2, and constant heavy chain 3, or some cases 4. Actually this is the constant heavy chain 2 which has the capability to activate the complements. So complement activating part of the antibody is part of the FC fragment. So FC fragment should remind you that that this portion crystallizes, this portion is having carboxyl end of the heavy chain, and if this antibody is going to activate the complement, then FC portion will activate the complement. And you will know later that IgG class of the antibody crosses placenta. IgG class of the antibody from the mother through the placenta goes to the fetus. So actually with the placental cell, this portion of the antibody bind and help the antibody to cross through the placenta. So it should remind you also about cross through, yes, placenta. And antibodies bind with specific type of cells. For example, later on you will learn IgE specially bind with catches the mast cell. Is that right? So again, IgG class of antibodies will catch the mast cell or bind the mast cell with this portion. So catching cells. Am I clear? No problem. So FC portion stand for what? Originally standard for this fragment could crystallize, but this should also remind FC stand for carboxyl end. FC stand for fragment which activate complements, fragment which help the antibody to cross the placenta, and fragment of the antibody which help it to bind with the cells. Any question up to this? And FAB portion, it's enough to say that this is a fragment which is antigen binding fragment. Am I clear? No problem up to this. Okay. Then, so we can say that one antibody has what thing? Antibody plus papain, yes, goes to what thing? Antibody will go into what? Antibody treated by papain will go into 2 FAB fragment plus 1 FC fragment. Is that right? Now we do another treatment. Take an antibody molecule, okay, let's make some seductive color scheme. Is that right? This is a typical antibody molecule, of course, consisting of 2 heavy chains and 2 light chains. Is that right? Now, rather than papain, this time we treat it by pepsin. You have heard of pepsin? What is pepsin? It is also proteolytic enzyme, not a name of a girl, pepsin, right? When antibody is treated by the pepsin, at which point pepsin will digest the antibody? It produces multiple digestions at this point. So actually now, when antibody is treated by the pepsin, then FC portion is digested at multiple fragments, but both FAB portion are still kept together. The important point is that pepsin attack the antibody molecule not at the hinge region, rather distal away from the hinge region towards the carboxyl end of the heavy chains. Is that right? Due to this reason, both FAB portion are kept together. Am I right? Now you will help me rapidly. If I say this is one antibody molecule and this is another antibody molecule, let me take your test. Enzyme attack here and divide the antibody molecule into how many fragments? 3. Which enzyme is this? Yes? Papain. And if I say that enzyme attack here and both FAB portion are still held together, what is this? Pepsin. Am I clear? No problem up to this? For sure? Okay. So now we have learned something about the basic structure of the antibody. Another thing, then when antibody is reacting with the antigen, when antigen and antibody bind together, they are held together by what type of forces and what type of bond? You know, when they come together, they make a bond, isn't it? So what type of bond is present between the antibody and antigen? Yeah, I'm about to be impressed by you. Okay, let me make our original molecule again. I hope you remember what are these chains I'm drawing? Heavy chains. And what are these chains? Yes? Light chains. And rapidly tell me, what are these bonds I'm making? Disulfide bonds. That's good. And what is this fragment? Variable of the heavy chain. And this is also variable of the heavy chain. And what should be here? This area? Variable of the light chain. And what should be here? Variable of the light chains. Then let's suppose I'm going to make the antigen. If antigen is like this, right? Now this antigen is if going to fit here, then what will happen? What was this area? Constant or variable? Yes? Variable. Now when this will fit into this, I think I have to make this a little lengthy so that it should look more proportionate with the yes. Now this is caught over here and right. So we say antigen is caught. It's caught in between the variable region of heavy chain and variable region of the light chain. This is what you already know. Now I know a little more. Actually, within the variable region, there are very special areas which make the fitting of variable region more better with the antigen, and these areas are given an additional name. You know, this is making a better fit, isn't it? These areas are called hypervariable regions. What are these called? These three are called hyper variable regions. So we say within the variable region of the light chain and within the variable region of the heavy chain, there are small amino acid groups, right, which are called hypervariable regions, which make the binding between the you can say antibody and antigen as precise as possible, very very precise. Is that right? Now I will draw the same thing here and you will tell me what's going on here. Yes, now you will tell me that this is variable of the heavy chain and this is variable of the light chain, and antigen is caught over right. But within the variable of the light chain, what is this region? Hyper variable regions. Is that clear? One thing which is very important to know, that in a given antibody molecule, the antigen will bind on the same side, one side, that is the same antigen is going to bind on the other side as well, right? So both sides have the same antigen specificity. Am I clear? No problem up to this? Right. Then I was talking about with which forces they are held together, or which type of bonds are in between them. What type of bonds are in between them? The forces with which antibody and antigen are interacting. Those forces are very simple forces. These forces are, yes, have you heard of electrostatic forces? Electrostatic forces, let me tell you how they are coming. For example, amino acid which is present over here is positively charged and amino acid here is negatively charged. They are attracted to each other. You see, they are held together by electrostatic forces or by the van der Waals forces. You know van der Waals forces? Van der Waals forces. So there are electrostatic forces or there are van der Waals forces. This wall is not that wall on which you sit and fall down, right? This is V. So electrostatic forces and van der Waals forces keep the antigen and antibodies together when they are specific for each other. And what type of bonds are formed in between? Is this ionic bonds or are they covalent bonds, right? Or they are like simple hydrogen bonds and hydrophobic bonds. Yes. Yeah, Dr. Safa is trying to tell. You know, the bonding between the antibody and antigen is, one way to tell is that is it strong bonds or weak bonds? Okay, only tell me this thing. They're weak bonds, right? If you bring, you know, let's suppose if there are, you bring lot of antibodies, right? Some of the antigens may dissociate and go to the other antibody if all the antibodies have same antigen specificity. So these bonds are not permanent bonds, they are somewhat reversible bonds or they are weak bonds. So we can say that these bonds are not ionic bonds, these are not covalent bonds, these are simply, yes, hydrogen bonds and hydrophobic bonds. So the forces which are keeping the specific antigen antibody molecule together are van der Waals forces and electrostatic forces, and making very weak bonds like hydrogen bonds and hydrophobic bonds. Am I clear to all of you? Is there any special question here? There is no. Okay, let's have a break.